Our next distinguished researcher is Dr. Nick Spitzer. Dr. Spitzer, I hope you won't mind me mentioning your anniversary here at UCSD. Dr. Spitzer has been at UCSD for, by my count, 41 years now. So you've reached the drinking age twice here almost. Dr. Spitzer, appropriately, Dr. Goldstein laid the groundwork for us, and Dr. Spitzer will talk to us now about the future of research in neuroscience. And please join us in welcoming Dr. Spitzer to the stage.
At this point, I had the opportunity to be drunk several times over, so hopefully I'll stay sober for the presentation this afternoon. I'm very glad to be here to talk about the future of neuroscience broadly and what it means for us here in San Diego. The future of neuroscience is unfolding very rapidly, much like that Larry has described for us with stem cells. A big part of it will be in neurotechnology. I'm going to try to focus in on this. I think it's going to be a technology that will be in many ways complementary to the technology that Larry's talked about. And as our last question brought up here, at the end of the day, this is going to be about companies, translation, patients, clinical trials. I think this is going to be happening here in San Diego. So this afternoon, I plan to touch on three topics in my remarks. The first will be to just sketch briefly for you some of the very exciting results that have emerged in neuroscience research, actually just during the recent few years. Then I'll talk at some length about the BRAIN initiative that I'm sure you've all heard about, the federal initiative, President Obama, and how San Diego was critically involved in actually promoting and launching this big presidential grand challenge. And I'll mention briefly how that's going to unfold nationally, but at the end I want to focus on what it is going to mean here at San Diego, what we're doing, and where we think this will take us. So when we think about the brain, it's a human brain here, we are immediately aware it's a very complex structure. Chatting with Bob earlier, he was quick to point out, even as a physicist, that this is arguably the most complex structure in the known universe. We have about 100 billion neurons, many more of those glia, including the astrocytes that Larry told us about. And each of those neurons has about 10,000 inputs. We call those synapses. These are the connections that Larry referred to. And so in this cartoon here, this very simple cartoon of the brain, we have processes like this axon coming down, making contact here with this dendrite, and this is, of course, the site of these synapses. So there are trillions, literally trillions of these synapses in each of your brains, and also in Larry's and in Bob's. So a lot of interest focuses on these synapses. They contain neurotransmitter molecules. This is on the so-called presynaptic side. They contain receptors for these molecules, because these transmitter molecules are released when an electrical signal comes flashing down the axon. And we're interested in these for two reasons, fundamentally. We talk a lot about plasticity, a topic that Larry's introduced for us this afternoon, initially with the wonderful remembrance of plastics as the appropriate career choice. Plasticity in this context means changes in the brain in response to changes in the environment or to experience. We would put in here, of course, learning and memory. This is one of the kinds of plasticity that we always want to keep fostering even after many years at UC San Diego. A second reason, then, in addition to the fact that the learning and memory changes occur here at the synapse, is that the neurological disorders that, again, Larry has alerted us to here, many of these disorders, not only Alzheimer's, but also schizophrenia, depression, disorders of this kind, are thought to result from malfunction at the synapses. So let me just tell you briefly about a couple of recent studies that have been going on at UCSD that highlight the very high quality and the impact of the work that's being done there. So here's a story, a recent story. This came out just earlier this month in this magazine, Nature. You know this magazine. This is a well-known magazine. And this is the work of Takaki Komiyama, a brilliant assistant professor on the campus now, only been there a couple of years. And it's looking at motor learning. So what's motor learning? Motor learning is what we do when we learn how to swing a golf club or to hit a tennis ball or even to ride a bicycle. Initially, when we start out, we make a lot of mistakes. And the trajectory of our swing is very uneven and it's very variable. But over time, maybe quickly, we become very adept at these motor behaviors. And our movements become highly stereotyped. And what Takaki and his colleagues have done here is to take a mouse, be able to peel away the skull, image hundreds of neurons in the motor cortex of the brain over a period of weeks when the animal is learning to perform a motor behavior. In this case, it's licking to get a drop of water. Mice are thirsty, just as you. And as they start to do this, the firing of the neurons is very heterogeneous, but over the course of just a short period of time, the neurons begin firing, certain constellations of the neurons begin firing in synchrony, and this is a result of the coordinated activity through synapses in the brain. So a fabulous physical insight about how many neurons are involved in this learning process, how they start to get together. In another study, again, very recent, in fact, it's so recent, published in Nature, that the citation has not yet appeared. This is only available on the internet. This is the work of Roberto Marano, a National Academy member there at UCSD, along with another Academy member, Roger Chen, a household name for us, a Nobel laureate. And they've been able to engineer a memory with long-term depression, LTD, and long-term potentiation. So how do memories occur? How are they actually produced? We've thought for a long time that it's a result of high-frequency activity at these synapses that changes the property of the synapses and produces this long-term potentiation. What they've actually shown now, and again, this is in mice, but presumably this is the way it's working in our brains, is that they could generate a memory. It was the memory of a tone that the mouse would hear, and in response to a foot shock, it would demonstrate a particular behavior. They could then abolish that memory by a particular kind of stimulation to produce long-term depression, and then they could reinstate the memory with long-term potentiation. And this was headlines wherever you're from. You may have seen this. This was a really dramatic demonstration of the physical substrate of memory in the brain. A remarkable accomplishment up here at San Diego. And finally, just briefly comment on some work that we've done looking at transmitter switching in the adult brain and how that regulates behavior. This was fun to do. This was a result of a lot of work over a sustained period of time, but with my colleagues, in particular Stefan Leutgeb, recently promoted, now associate professor at UCSD. Last year, we showed that in a rat, presumably the same thing will happen in a human brain, in response to different sensory stimulus. In this case, it was day length, photo period, long day, short day. Right now, we're enjoying the longest day of the year here in San Diego as elsewhere. This can change the identity of the transmitters in particular regions in the brain. This was a scientific heresy. This was thought not to happen. We thought that transmitters were fixed, unchanging, constant, invariant throughout the life of the organism. Turns out, not true. And remarkably, when that transmitter switches, so does the behavior of the animal. So I hope you're getting a sense here that there's a lot of very exciting neuroscience going on at UC San Diego in these areas, in addition to the areas about which Larry told us. So how did the Brain Initiative get launched? This is a wonderful story, and it's very much a UC San Diego story. This began in 2011, so not that long ago, with a meeting in England at Chichley Hall at the Kavli Royal Society International Center. This is a wonderful English estate, a fabulous place, and it was the brainchild of the Kavli Foundation thinking that it would be very important to bring together engineers, nanoscientists with neuroscientists and see what kind of cross-fertilization, what kind of interaction could arise in this kind of environment. They met for 3 days, they talked, they discussed, and they realized that these two silos, again coming back to Larry's metaphor here, actually had a lot in common, and that corn could talk to wheat, and that there was going to be an opportunity here to make things happen. Within a month, they had developed a white paper, the whole brain activity map, merging nanoscience and neuroscience for technology and health, and delivered it to the OSTP, the Office of Science and Technology Policy. So this was moving pretty quickly. Enthusiasm built very quickly over time, and by June of the following year, the Brain Activity Map project was now identified. The idea was to understand at a functional level how nerve cells are connected to one another. We're beginning to get a sense of how that works at the anatomical level. Of course, we want to understand how that works at a functional level. And this enthusiasm continued to build. Brain Activity Map, a nice report in Science in March of 2013. But even before this article appeared, talking about how one could make progress on this front, we were blown away in the State of the Union address in February of 2013 when Barack Obama, out of a clear blue sky, of course not quite a clear blue sky, here with Joe Biden and John Boehner, talked about the challenge of mapping the human brain. And so this was something that started with Ralph Greenspan, one of our faculty at UC San Diego, Terry Sejnowski at the Salk Institute. These two gentlemen were on the ground at the very beginning. I had the good fortune to join them shortly afterward and respond then to this federal challenge, this grand challenge of moving ahead to map the human brain. In April, so this is February, now in April, there were a group of us, Pradeep Khosla, our chancellor, Ralph, Terry, myself, and others. We were in the White House in the East Wing, and we were there to hear in more detail about the BRAIN Initiative. So here's Barack Obama. We all recognize him. Many of you will know Francis Collins here on the left, the director of the National Institutes of Health, and he's introducing the man whom he characterized and who was very proud to be the neuroscientist in chief. And so this was terrific. A lot of enthusiasm, a lot of public press on this point. And for us here in San Diego, it was terrific, as you'll see, because it led to something that's very tangible, very concrete, that I think will have lots of legs for us here in town. So what was the BRAIN Initiative? What is the BRAIN Initiative? In broad terms, it's on the order of about $100 million for fiscal 2014. This is with the objective here of doing some very good, very useful things to begin to understand how the brain actually works. It's phenomenal that after really a century of hard work on this problem, we are remarkably ignorant about that process. Many of you will be thinking, as we did too, this is actually small money because the support through the National Institutes of Health alone for neuroscience is on the order of $5 billion. That's with a B annually. So this is really a tiny fraction, but it's in many ways comparable to the 01 year of funding that was provided for the Human Genome Project. The challenge, of course, is figuring out how are we going to spend more money. One needs to begin to sort of seed the ground, get things started, find out where the money is going to be usefully placed. You know, how much money can we give to Bob? How much money can we give to Larry? And so on. So this looked like a very good start, and it's going to be distributed in the following forms. The Defense Advanced Research Projects Agency, DARPA, that many of you will be familiar with, is going to be the high roller here, coming in with about half of that amount. National Institutes of Health going to provide about $40 million, and then the National Science Foundation is coming in at the end here with $20 million. So a little over $100 million, but a very very good way to start the process here. And then there's going to be substantial support in this initial period and then continuing forward from a number of private supporters. So the Allen Brain Institute in Seattle is going to come in at $60 million a year for the next period. The Howard Hughes Medical Institute, HHMI, is going to be a very substantial contribution. Kavli Foundation is going to contribute in proportion to its ability to do so. And the Salk Institute has ponied up at a very nice level here to provide support for this brain initiative. Importantly, and Larry referred to this in his remarks, it's very important in these large efforts to focus also on some of the ethical issues that arise here when we come to work on the stem cells on the one hand or the brain on the other hand. These are important issues that go to the core of the health and the identity of individuals, and so it's important to consider that very thoughtfully. But I hope you're noticing what's at the bottom of the slide here, because this, in our minds, is almost certainly the major driver in Barack Obama's mind. And this is the economics of this brain initiative. Let's just remember what the story was for the Human Genome Project. This was a $3.8, roughly $4 billion investment over a period of time. And the remarkable thing that came about, and this was the result, this was analyzed, the results were analyzed by the Battelle Institute. They studied the economic impact of each dollar that was invested in the Human Genome Project. And it turned out there was an enormous multiplier, I think about $141 of economic return for every $1 invested. So this is very impressive historically. It's been great, of course, for the country. And I'm pretty sure that Barack Obama had this in his mind when he thought about the Brain Initiative, and I'll come back to that a little later. So, how to spend this money? The National Institutes of Health went about this in a very thoughtful way, and I want to highlight this for you in a very particular way. They put together a blue ribbon committee that over the course of the last year now, since April of 2013, has been studying and thinking and meeting and consulting with experts on how to best deploy this money, what kinds of questions should be the number one questions, what questions are the number 2, 3, 4, and questions. And they came up very recently now, June 5th, with their report. It's the composition of this committee that I want to highlight for you. Here are the 15 individuals, and I've framed here in light green the 6 of the 15 that are from the state of California. So California has a very big stake in this process. 3 of these individuals are from Stanford, 1 from Caltech, and 2 from San Diego. And we've already mentioned them, Roger Chen and Terry Sosnowski. So California has a big stake in this process and is already influencing the direction of this spending of this money, and I think it's going to turn out to be a very useful thing for San Diego. So let's think for a moment about the large initiatives that we've had in this country, national initiatives. All of you, I think, will remember, perhaps many of you were around at the time, the declaration by John F. Kennedy in 1961, we're going to put a man on the moon. This was a visionary challenge. It was a very ambitious challenge at the time. Nobody knew how to do that. But, of course, it delivered the goods. A little later on, a decade later, we had the war on cancer. And that was harder to achieve. I think the war was fought, as you heard from Larry. It continues to be fought. It's not a war that we're going to win anytime really soon, but we're making good progress on that. And importantly, the efforts of the war on cancer catalyzed an enormous amount of progress tool development in the biological sciences. And that, I think, should be counted as a real win in the war on cancer. More recently, as I've mentioned, we had the Human Genome Project, 15-year project. And again, we've been able to sequence some genomes, and more of that is happening, as you know, and as we heard. Again, much of that success of that period was in developing new technology that has been very helpful to us. So finally, here we are with the 2013 BRAIN Initiative. What is it about this initiative that is likely to lead it to be successful at the very high level that we anticipate and want from a national challenge? Well, I think that the first thing to point to is that it really is a visionary goal. It is a staged goal. One is going to reach this goal in a series of steps, and the NIH study group has outlined what those steps should be in their view. I think it makes a great deal of sense. I'm not going to take you through that this afternoon. But a lot of the goal involves trying to understand at a very high level how neural circuits work, how individual circuits that are involved in motor activity, for example, or in decision-making, for example, how these circuits actually work in the brain. These are visionary goals. Second, the timing here is really almost perfect. And the reason for this is that the engineering silo and the neuroscience silo have suddenly realized, starting with that meeting at the Churchley Hall in the United Kingdom in England, they've realized that there are real opportunities here. The bioengineering, as you'll hear from Shu Chen, nanoengineering, electrical engineering, computer science and engineering, these have all in the past 10, 15 years come to a very interesting and very high level of sophistication that can now be extremely useful to neuroscience. On the other side, the neuroscientists have realized, embarrassingly, probably for the first time, that they can really take advantage of these new tools and accomplish things with them that they just dreamed about in the past, didn't have the technology. The objectives have to be focused. You have to spell those out. I've mentioned that already. But finally, I think economics, again, is going to be something that is going to really carry the day here. I think the vision is that there will be, as I'll describe shortly, ways in which this will develop new technologies, and those technologies will have high impact on the country nationally, but we hope very specifically here on San Diego. So let me tell you about Moore's Law for neurons. Now, you're all familiar with Moore's Law. Moore's Law states that the number of transistors on a chip will double every 2 years. And this was a prediction that was made now many years ago, but it's been amazing the way in which Moore's Law has stood up over time. And so what we've plotted here are the numbers of neurons in the brain that can be recorded simultaneously as a function of the calendar. And what you can see is that back in 1960, we could record, on this semi-log plot here, we could record a couple of neurons at the same time. And I was a graduate student back then, and we thought that was pretty cool. But you're remembering now that we have 100 billion neurons in the brain, so it's kind of small potatoes when you think about it. And then over time here, we've seen that the numbers of neurons that can be recorded simultaneously has increased, and this is good. But notice how good it is. Not very good, right? This is only about 300, maybe 500 in a good day, in a good system. And so when we think about what our goal is, let's just think about recording from 1 million neurons. At this rate, we'd get to 1 million neurons here, 10 to the 6th, at the end of the century. Now, that's really not acceptable. That's just too slow, right? That's just embarrassing. And so the plan is now to speed this up so that in 15 years, we can at least get to 10 to the 6th. Now, some of you are going to be thinking, well, come on, Nick, 10 to the 6th, you mentioned 10 to the 11th. Yes, true, but if we can record from 1 million neurons that we can select, and we can record from neurons that are in a particular pathway, that are subserving a particular function, a particular behavior, then we think we can really understand how those behaviors are generated generated. So this is very exciting. Let me, in the last part now of my presentation, talk a little bit about what's happening here in San Diego. And very nicely, we've been able to establish, thanks to many friends, Bob Dines among them, a Center for Brain Activity Mapping here at UC San Diego. This is something that Pradeep Khosla actually enabled within 6 weeks after the April 2013 meeting in the East Wing of the White House. And my colleague Ralph Greenspan is the director. It's part of the Kavli Institute for Brain of Mind that I direct. So Ralph and I working hand in glove on this process along with Terry Sejnowski. And we've had a series of objectives that have played out now rather nicely in the year that we've been in business. The first thing that we did was to attack front on the engineering silo and the neurosciences silo and find ways to melt the walls so that these people would begin seeing what the opportunities were to talk with one another. And we did this by getting together at the Qualcomm Institute. It's a wonderful venue, and we had tables and whiteboards and people. We had engineers pitching to neuroscientists, and then we'd have another one with neuroscientists pitching to engineers. And I have to tell you, the engineers had developed these amazing little widgets that could do things that would blow your mind, and they really didn't know how they were going to apply them. They didn't know what to do with them. The neuroscientists, on the other hand, were hungry for technology that would allow them to begin to record and stimulate from large numbers of neurons all at once. So these conversations were very exciting and got people thinking about how can we collaborate? How can we work together? How can I as a neuroscientist use your tool as an engineer to achieve some of these goals? This then led to the award of seed money grants here in San Diego. Pradeep Khosla provided $1.5 million to launch this process. These are small grants, really seed money grants, but they provide the opportunity to put an engineer and a neuroscientist in the same room, lock the door, wait for the white smoke to come out, and see what can happen. And we've funded now about 24 of these innovative seed money grants, and I'll just illustrate for you some of the topics of these proposals. You get a feeling for what's being funded, what these guys think that they can do. So here's a fully encapsulated wireless, modular, and conformable neural recording system that should be able to record and wirelessly, while the animal behaves, moves about, beam the input and multi-channel input back to a computer. Here's another kind of completely different technology involving nanoshells. These are tiny little micron, sub-micron sized plastic biodegradable shells that contain enzymes that report on neurotransmitter distribution in the brain. We've already talked about synapses and how neurotransmitters are key to the function of synapses. You need to know what neurotransmitters are where, when they're being released, what their distribution is. So this is a very exciting project. Some of you will remember, now this was a long time ago, this certainly dates me, the movie The Fantastic Voyage. Anybody remember that? Anybody? Yeah, good, okay, right. So this was a wonderful fantasy at the time involving human beings that were shrunk down to sub-micron size, got into a little submarine that could then travel through capillaries throughout the entire body and be able to report on the status, healthy, unhealthy, of different organ systems. Well, it turns out now that technology can probably arrive. It's possible now to make very small robots that can travel through blood vessels, that can be recording the activity, the chemical, the electrical activity in the brain, and also can be recording activity in the liver and in the pancreas and in the lungs and in other tissues. So this is going to be an amazing technology when it comes to reality. Another kind of approach has been to have these micro pillar arrays of very fine needles that could be pushed into the brain without damaging the nerve cells in the brain to be able to record from very large numbers of cells in 3 dimensions. And finally, optical methods are being developed that will allow through focus capture of the activity of neurons in the intact and functioning brain. So these are just a few of the kinds of seed money grants that we're funding with the Center for Brain Activity Mapping. And then finally, we've convened scientific meetings to actually bring together people from across the country to rapidly disseminate progress. The first of these was again held just this month here in San Diego, June 4th. This was a symposium on sensor technologies for the nervous system. What are the technologies currently available out there that people are developing, that people are using to record the activity from as many neurons as possible in the brain? Speakers including people from MIT, from Harvard, from the Scripps Research Institute here, Sanford Burnham, from Santa Barbara, from Chapel Hill, across the country here, coming to San Diego. Very exciting symposium, lots of interest and lots of further collaborations set up as a result of this process. So this is locally in San Diego. Some of you will have followed perhaps the passage of the California state budget, thanks to Ralph Greenspan's efforts, and again with great political input both from Larry Smarr, from Bob Dines, and also from Dick Atkinson. This really was very well positioned, and we saw the creation of Senate Bill 836, brain research, a cow brain program. So this is not the federal brain initiative, this is the California initiative, Cow Brain, state of California. And at the time that I prepared these slides, all I could tell you was that the budget of $2 million for fiscal 2014 was going to be voted by the legislature on June 15th. That had happened. But very nicely, just last Friday here in San Diego, Jerry Brown, our governor, signed into law this very nice $2 million for CalBrain. So this is now a little out of date. It's not on the governor's desk. It's beyond the governor's pen. And this is going to be spent now to jumpstart a program that will be across the state of California. This is not all going to be based in San Diego, but we believe that this would be the initial funding, just like the initial funding for the Brain Initiative, that will then lead on next year if, as Bob pointed out, the economy in the state of California continues as strong as it is, to allow us to achieve even greater support from the state. And here's just some language to indicate what was going on in the Senate bill. The Brain Initiative has the potential to be a major driver of new industries, jobs in biotechnology, artificial intelligence, information technologies, as well as a catalyst for major breakthroughs in brain-related diseases, injuries, and illnesses of the kinds that Larry Goldstein told us about. So what's the plan for San Diego? We think, and I hope I tried to give you some flesh on the bones here, we think that innovative neurotechnology will be developed here in San Diego. Yeah, it'll be developed elsewhere, but we think we're way ahead when we talk to our colleagues around the country with what we're getting to happen here. We think that as a result of that, companies will be founded. That was the pattern for the Human Genome Project, a number of companies founded here in the San Diego region. We think that will happen for neurotechnologies as people develop these amazing tools. They want to manufacture these tools. They want to disseminate the sale of these tools to neuroscientists and engineers around the world. We think that will happen here in San Diego. One thing that's for sure to happen is that we will be training students. This is part of the educational mission that Bob Dines talked about. Students that are cross-trained in engineering and in neuroscience, so they can bridge this, what used to be a divide, and now work seamlessly across these different fields. And so finally, there will be jobs here in this sector in San Diego. So we're very hopeful here for a very bright future for this process. I want to close then with just a quick shot. Some of you will know this view. This is the view of the shore, just a couple hundred yards to the west of UC San Diego. Wonderful spot, but I want to highlight here in my acknowledgments to Ralph Greenspan, Terry Sejnowski. I've mentioned their names a lot. Also Chancellor Pradeep Khosla, been a real engine here for us driving this process, as well as UC President Emeritus Bob Dines and UC President Emeritus Dick Atkinson. Thanks very much.
I think we have time for one question for the doctor. Anybody?
Yes, the question is, what's the relationship of the BRAIN Initiative and CalBRAIN and Center for Brain Activity Mapping to the European BRAIN Project, which is a very big project, very well funded. This is a wonderful project, very complementary to what's going on here in the United States. If I can characterize it briefly, the European BRAIN Project is involved in taking existing data, both functional and structural, anatomical, about the brain, and existing computational algorithms, and trying to reconstruct how the brain works, how a small piece of the brain would work. We think this is a very important goal. What we're doing here is trying to acquire additional data with these new tools and develop new computer algorithms to go beyond that. We will build, in part, on some of the things that the European Brain Project have already developed. Great question.